When most people think about wetlands, their mind probably goes to muddy water and mosquitoes. But when I think about wetlands, I'm seeing something completely different. I see essential ecosystems that are filtering our drinking water, protecting from flooding, and supporting a variety of wildlife species. Unfortunately, wetlands are one of the most threatened habitats on Earth. In the United States alone, we've already lost more than half of our wetland habitats. And as we continue to see a loss of these wetlands, we're in return seeing a loss of the plants and animals that depend on them. But protecting these plants and animals requires more than just protecting the habitat they live in. Hi, my name's Beth. I'm a graduate student in the Department of Plant Biology at the University of Georgia, and my research is using genetics to answer a really important question in conservation biology. How can we protect rare and endangered species before they're lost? One of the species I work with is the Georgia sweet pitcher plant. This carnivorous plant is found in a handful of wetlands only here in the state of Georgia. And yes, they really do eat insects. Growing in nutrient-poor bogs, they've evolved this incredible adaptation to make up for what the soil lacks. Despite its unique biology, the Georgia sweet pitcher plant faces an uncertain future. Although it was recently identified as a species of greatest conservation need in Georgia's State Wildlife Action Plan, it still lacks state or federal protection. As populations continue to disappear, we still lack the information needed to protect those that remain. To develop effective conservation strategies, we first need to understand population health. At first glance, that might seem as simple as counting how many plants are left, but that really only tells part of the story. Above ground, two populations could look exactly the same, yet one may be genetically healthy, while the other is slowly losing the genetic diversity it needs to survive. Genetic diversity is the variation in DNA within a population. It's what gives populations the ability to adapt to environmental change, resist disease, and survive over time. In my research, I measure genetic diversity across a species range to identify healthy populations and those that are most in need of conservation. This kind of data-driven conservation planning helps us make better decisions and direct limited resources where they can have the greatest impact. But my favorite part about this work is that it doesn't just stay in the lab. Whether I'm working with state agencies to inform conservation planning and status assessments, botanical gardens to safeguard at-risk populations through conservation collections, or nonprofits and volunteers working to restore native habitat, every conversation is an opportunity to connect science with action. Because the best research doesn't end in publication.
It begins with conversation.